The JAK-STAT signaling pathway, pharmaceuticals, side effects, and plant-based compounds. Cytokines are small proteins or glycoproteins with a mass range of 6 to 70 kilodaltons. They are involved in intercellular signaling, regulating a variety of physiological processes, including differentiation, proliferation, survival, and apoptosis of their target cells, as well as immune responses and inflammation. Cytokines can be released from cells of the innate and adaptive immune systems, as well as other cells of the body, such as adipocytes, astrocytes, fibroblasts, and endothelial cells. The effects of cytokines are mediated by specific receptors on target cells. Cytokines are extremely diverse. They can be divided into several protein families, including the tumor necrosis factors, TNFs, interleukins, ILs, interferons, IFNs, colony-stimulating factors, CSFs, and transforming growth factors, TGFs. Cytokines are instrumental for the maintenance of healthy tissues and the upkeep of a well-functioning immune system. However, elevated cytokine signaling may result in autoimmune disorders, inflammation, and oncogenesis, while insufficient signaling may increase the body's susceptibility to infectious diseases. A high percentage of the population suffers from chronic inflammatory skin diseases. These non-communicable diseases are caused by genetic and environmental factors, as well as autoimmunity. Examples of chronic inflammatory skin diseases are atopic dermatitis, psoriasis, alopecia areata, and chronic spontaneous urticaria. Atopic dermatitis mainly affects children. Its pathogenesis can be attributed to skin barrier dysfunction, immune dysregulation, and microbiome. Often there is an increased burden of Staphylococcus aureus, contributing to inflammation. Psoriasis is a chronic T helper cell-mediated inflammatory skin disease that can also affect other organs. Many drugs have been developed and approved to treat plaque psoriasis in particular, although the disease can manifest in many ways. Alopecia areata affects approximately 2% of the global population. It is an autoimmune disease of the skin, where T cells attack hair follicles, resulting in inflammation and hair loss. Chronic spontaneous urticaria is a mast cell-driven autoimmune disease that presents with itchy, raised and swollen areas on the skin or angioedema. Responsive patients can be treated with antihistamines. Inflammatory conditions of internal organs include bowel diseases such as Crohn's disease and ulcerative colitis, joint diseases like rheumatoid arthritis and psoriatic arthritis, and asthma. Crohn's disease, ulcerative colitis, and arthritis are autoimmune diseases, and asthma is an inflammatory disease triggered by allergens, pathogens, or other environmental factors such as smoke. While ulcerative colitis is mainly a disease of the colon and the rectum, Crohn's disease can affect the entire gastrointestinal system. These inflammatory bowel diseases are caused by immune attacks against components of the gastrointestinal tract that may be triggered by genetic predisposition and environmental factors such as infection. Rheumatoid arthritis leads to gradual joint deterioration. It can be linked to genetic predisposition and environmental factors such as smoking. Psoriatic arthritis is a chronic inflammatory disease of the joints often associated with psoriasis. Both the innate and the adaptive immune systems are involved in the pathogenesis of rheumatoid or psoriatic arthritis. Both forms of arthritis are risk factors for cardiovascular diseases. Inflammation may lead to neoplastic diseases and progression to cancer. Polycythemia vera and myelofibrosis are hematopoietic stem cell disorders associated with inflammation, increased cytokine levels, and a mutation in the Janus tyrosine kinase 2, JAK2. Janus tyrosine kinases, JAKs, are cytokine-dependent cytoplasmic enzymes that catalyze the phosphorylation of STAT proteins, which subsequently enter the cell nucleus, where they regulate gene transcription by targeting promoters. JAKs are large proteins with unique structures. Their location in the cytoplasm is near the membrane. Upon ligand binding to extracellular domains of a transmembrane cytokine receptor, JAKs are brought into proximity with each other, resulting in the phosphorylation of themselves, the receptor, and signal transducer and activators of transcription, STAT proteins, which then dimerize and translocate to the nucleus. Many cytokines, including ILs and IFNs, exert their effects via Janus kinases, of which there are 4 types, namely JAK1, JAK2, JAK3, and tyrosine kinase 2, TYK2. JAK-mediated physiological processes include hematopoiesis, the regulation of immune responses and inflammation, tissue repair, apoptosis, and the formation of adipocytes. However, JAK-mediated processes also play an important role in malignancies and autoimmune diseases, which has warranted the need for pharmaceutical intervention. Potential pharmaceuticals may target cytokines, cytokine receptors, JAKs, or STAT proteins. Tocilizumab, sarilumab, siltuximab, ustekinumab, and mepolizumab are examples of cytokine or receptor antagonists. Tocilizumab and sarilumab are monoclonal antibodies that neutralize interleukin-6 (IL-6) receptors, whereas siltuximab targets the cytokine interleukin-6 itself. A major clinical application for these antibodies is the treatment of rheumatoid arthritis. Ustekinumab is a monoclonal antibody that targets interleukin-12 and interleukin-23, and it is used for the treatment of plaque psoriasis, psoriatic arthritis, Crohn's disease, and ulcerative colitis. Mepolizumab, a monoclonal antibody targeting IL-5, is used to treat severe eosinophilic asthma. Biological agents like these are the standard care for many immune disorders. However, they need to be administered parenterally and have pharmacokinetics that are variable and difficult to predict. Another issue is that patients may produce antibodies against these agents. JAK inhibitors are small molecules suitable for oral application. Examples of these drugs are upadacitinib, abrocitinib, fedratinib, ruxolitinib, baricitinib, and tofacitinib. Upadacitinib and abrocitinib are selective JAK1 inhibitors. Upadacitinib is used for the treatment of conditions such as rheumatoid arthritis, psoriatic arthritis, atopic dermatitis, and ulcerative colitis. Abrocitinib has atopic dermatitis as its indication. Fedratinib selectively inhibits JAK2. It is used to treat myelofibrosis. Ruxolitinib and baricitinib are inhibitors for both JAK1 and JAK2. Ruxolitinib is used for the treatment of myelofibrosis, polycythemia vera, and graft versus host disease. Baricitinib may be used to treat rheumatoid arthritis. Tofacitinib is a non-selective inhibitor blocking JAK1, JAK2, and JAK3. It is approved for the treatment of rheumatoid arthritis, psoriatic arthritis, and ulcerative colitis. Deucravacitinib is the first tyrosine kinase 2 inhibitor with approval for clinical use. Its primary indication is plaque psoriasis. Selective JAK inhibitors are expected to have a better safety profile than non-selective ones. However, even if they inhibit only one of the 4 JAKs, they will inhibit signaling pathways for a large number of different cytokines. JAK inhibitors, while very effective, can have severe side effects. Adverse drug reactions include infection, hematologic abnormalities such as neutropenia, lymphopenia, thrombocytopenia, and anemia, cardiotoxicity including cardiovascular death, and malignancies. Safety warnings have been given in many regions, including the USA, Canada, Europe, Australia, and New Zealand. The STAT protein family has 7 members with the following major functions. STAT1 and STAT2 regulate immunity against viral and bacterial infection. STAT3 regulates innate immunity, inflammation, stem cell maintenance, and cell metabolism. STAT4 regulates the development of immune cells. STAT5A and STAT5B regulate the development of multiple immune and blood cell lineages, and STAT6 regulates innate and humoral immunity. While STAT3 signaling is necessary for cell maintenance, increased activity promotes pathological cell proliferation and survival, and can lead to tumor development and progression, and it is continuously activated in several types of cancer. STAT3 has therefore become of considerable interest as a potential drug target. Clinical trials have been conducted, and many drug candidates tested have shown excellent anti-tumor activity. However, due to issues with safety, stability, and bioavailability, no STAT3 inhibitors have received market approval to date, October 2025.
Many plant-based compounds have also been found to modulate the JAK-STAT pathway. Some examples are listed below. Hypericin, mainly found in Hypericum perforatum, St. John's wort, has been suggested to be a JAK1 inhibitor based on in silico analyses. It has anti-tumor, antiviral, and neuroprotective properties. Dry extracts of Hypericum perforatum are available as phytopharmaceuticals for the treatment of depression. Amintoflavin, which is contained in ginkgo biloba, has been shown to have anti-inflammatory, antimicrobial, anti-cancer, and neuroprotective activity. In silico analyses have suggested that amintoflavin is a JAK2 inhibitor. Ginkgo biloba dry extracts are available as phytopharmaceuticals and are used in patients with decreasing mental capacity. Arbutin, a polyphenol originating from Arctostaphylos uva ursi, commonly known as bearberry, has been shown to ameliorate colitis through JAK-STAT pathway inhibition. Bearberry leaves are available as tea or their dry extract as phytopharmaceuticals to treat uncomplicated urinary tract infections in women. Quercetin is abundant in numerous dietary sources, such as onions, apples, berries, capers, broccoli, grapes, tomatoes, nuts, honey, and tea. It too inhibits several components of the JAK-STAT pathway and possesses anti-inflammatory, anti-allergic, antiviral, anti-cancer, and anti-diabetic activities. Citrus reticulata, commonly known as mandarin, is the main source of 5-dimethylnobiletin, which has anti-inflammatory, antioxidant, antimicrobial, neuroprotective, and anti-atherogenic properties. An in vitro study reported the repression of JAK2 and STAT3 expression by 5-dimethylnobiletin. Psoralen, which is contained in citrus limon, the lemon, has been reported to have anti-tumor activity by inhibiting JAK2. Lycopene, predominantly found in tomatoes, has been shown to have antioxidant capacity and broad anti-cancer activity. It has been indicated that lycopene suppresses Helicobacter pylori-induced JAK1 STAT3 hyperactivation in gastric epithelial cells and can therefore prevent Helicobacter pylori-associated carcinogenesis. Mangiferin, a polyphenol found in mango, has been shown to reduce the phosphorylation levels of JAK1, STAT1, and STAT3 in gingival epithelia in the mouse model, indicating that it may be beneficial to treat periodontitis. Green tea has many health benefits, largely due to its main active ingredient, epigallocatechin 3-gallate, which modulates several steps of the JAK-STAT pathway. In addition to blocking the phosphorylation of STAT1, epigallocatechin 3-gallate also suppresses the activity of interferon gamma, JAK1, JAK-2, and STAT3. Studies have reported antioxidant, anti-cancer, anti-diabetic, anti-inflammatory, and antimicrobial activities of epigallocatechin 3 gallate.